author of The Processes of Life, A Brief Introduction to Contemporary Molecular Biology for the Seriously Curious, published by MIT Press. He's the director of the Computational Bioscience program at the University of Colorado School of Medicine and a professor of pharmacology there. He got his PhD in computer science from Yale University in '89 and was immediately hired as one of the first computational staff scientists at the NIH. He also holds a professorship in computer science at CU Boulder and has an avid interest in both bioethics and scientific art. And he's also a biker. And tomorrow he's going to be riding 85 miles, and it's the first organized ride that he's done. So everyone should support him in that by thinking good thoughts.
It's a cancer fundraising ride, so if you want to donate, I can come up with a website where you can donate. It's a 2-day ride, and Sunday we do 65 in the foothills outside of Fort Collins. So.
There's more. Welcome, Larry.
Thank you. That's a very nice introduction. And despite what the intro said, I'm happy to take questions in the middle. If I said something that confused you, wave your hand, and you probably weren't the only one that was confused. And I'm going to start off by admitting that I made a mistake on my very first slide. That's not my email address or my website. Those have both been dead for years. I don't know how that got on there. Substitute for UC-HSC, UC Denver, and it will work. Our university has branding issues. Sorry about wandering away from the camera. And I'm told that you guys like to tweet, so my Twitter handle, which is also usually on the title slide, is Prof. L. Hunter. So feel free to comment or follow. But let's get started. So you probably know or have heard all the interesting stuff going on in molecular biology these days, in genomics, the human genome sequencing, and all that kind of stuff. And that has really changed dramatically in the last 10 years and is going to change dramatically again in the next 10 years. We're in the middle of a really exciting time in science. And mostly the reason for that is technical. And this graph shows you, is there a pointer, by the way, or should I'll just wave. This graph shows you why. And this is a logarithmic graph. Up in the top left corner, you'll see $100 million. And that's what it cost to sequence the first human genome in 2001. And then down towards the bottom right, you see $1,000. Okay, so this falls logarithmically or exponentially, which is what it costs to sequence a human genome now. All right, so in a little over 10 years, we've gone from $100 million to $1,000. Okay, much cheaper. Okay, I can afford one and not the other. And you can see that line, the white line on the top there, is Moore's Law line. That's the price or the rate at which prices of computers, or actually density of transistors, but pretty much the same thing, has come down. Okay, and so the price of computers has cut in half pretty much every 18 months since the 1960s. And that has gotten to the point where the thing I carry around in my pocket has more compute power than a supercomputer did when I was in college. All right, so that's what's going on in genomics, except we're in the early stages of that. This is for the computer equivalent to the 1960s or something like that in genomics. All right, we're just getting off the ground. And this ability to sequence genes or sequence DNA has applications in lots of places other than just sequencing genomes. So our ability to read DNA allows us to peer into the inside of living things in ways that we have never been able to do before. And you'll hear some more in detail about that from Robin when I'm done. But I'm going to try and give you an introduction to sort of what this means and what the information resources are. And I'm a huge fan of librarians, and I want to give you both some intuitions about what you ought to be telling people, and because people's intuitions about genes are terrible for the most part. Almost everything you think you know about genetics is probably not true. And also to sort of point you at reliable information resources, because there's an awful lot of unreliable information resources out there about genetics. My favorite example, the FDA recently, about 2 years ago actually, shut down a whole bunch of the direct-to-consumer genetic testing companies, gave them all warnings and shut a bunch of them down. Because the majority of the companies that were directly marketing genetic tests to consumers, you'd send in your cheek swab or whatever, and they'd claim to sequence it and then claim to come up with some nutritional supplement that would help you lose weight. Okay, and they were mostly throwing the samples away and selling the same supplement they always sold, and there's a lot of that going on. So we really do need to pay attention and provide reliable resources about genetics to the broader community. Scientists need your help. The lay public needs your help. Sick people often are the ones who come to find out about genetics, and they especially need your help. So with that, let's get started a little bit. This genetic information has really changed a great deal about what we understand about life and how medicine works. So I want to show you a little bit about both of those things. So on the, let's see, your left, wish I had a pointer. Let's see, can I use the, here we go. Okay, so over here, starting around the same time we got the first human genome sequence, we identified the first gene relevant to a common human disease. So we already knew about some rare genes that usually show up at childbirth that created disease. But this one, a gene called PPAR gamma, is related to diabetes risk. And so that was the first one found in 2000. And then there was kind of a dribble of them, and you can see the diseases. These are common diseases. And then the sluices really opened up in the middle of the last decade. And this is what it looks like now. Each one of those dots is a gene where there's a variant that plays some role in some common disease or other phenotype. So diseases loosely here. One of those is sneezing when you walk out into bright sunlight. Turns out there's a genetic variant for that. Some of them are actually really important in medicine. So down here I grabbed a couple of headlines. These are both from a few years ago now, where this one is about warfarin dosing. Okay, so warfarin is a very commonly given drug. If you have a clotting problem, like you got phlebitis from flying out here and sitting in an airplane too long, we'll usually give you warfarin. Warfarin's an anti-clotting drug. It's actually rat poison, but also an anti-clotting drug that comes with a very narrow therapeutic index. What that means is the effective dose is awfully close to a dose that is bad for you. So we have to get it just right. And so the way we usually do that is we start you off on 5 milligrams and see how you do. We test your clotting, and if you're not responding well enough, we give you more. And if you're bleeding, we give you less. And that mostly works. Except for a few people who are very sensitive to warfarin, and they get very bad bleeds, and some of them have very bad consequences for that initial dose. So it turns out we can identify a lot of those people genetically. We can pick out the ones who are going to be hypersensitive to that drug by giving them a genetic test. And there's actually two ways that people can be hypersensitive to the drug. One is they can be very slow metabolizers, so the drug stays in their body longer than it stays in other people's. And the other is they can react, the place where the drug does its work, and the vitamin K receptor, turns out to be hypersensitive to the drug. So those are, the first one is pharmacokinetic, how fast the drug moves through your body, and the other one is pharmacodynamic, how the drug works in your body. And so that turns out to be really important, and the FDA has recommended that you give genetic tests to everybody before you give them warfarin, just to pick up those relatively few rare people who would have a bad consequence of that first dose. And it has not been widely picked up by the medical profession yet. It'll probably happen. Somebody will sue, and then everyone will do it. But for the time being, it's an FDA recommendation. There's another one. This is clopidogrel here is probably better known as Plavix, a very widely prescribed drug also for people with heart disease. And it also has a genotype that is, in this case, it's a very fast metabolizer, so the drug doesn't work very well for you. So we have to give you a bigger dose because you eliminate it very quickly. So we're starting to see these applications of genetics in real day-to-day medicine for most people. It's not an obscure backwater anymore. It's really changing the practice of medicine, and that's going to happen in a much bigger way coming soon. And it also changes our understanding of life. So, for example, one of the discoveries that was made here, and a lot of the work that's done here, is about the importance of your microbiome. How many of you have heard of the microbiome? Oh, good. All right, half. So the microbiome is all of the bacteria that live around and on you. It turns out you have more bacterial cells than you have human cells, most of which are in your gut, but a lot of them are in your skin or in your nose and your ears and stuff like that. And it's fairly stable, and it has a lot to do with your health and disease. And until recently, we only knew about a few of those bacteria. You've probably all heard of E. coli. That's one of the ones that lives in your gut. But the ones we heard of were the ones that grew like weeds when you put them in a petri dish, right? Give them sugar and they grow. But that's not most of them. Most of them won't do that. You put them in a petri dish and they die. So we didn't know about them until we had this DNA technology that we could use to go figure out what's in there. And so it's been huge. So we understand a great deal about life and disease and all this other stuff based on this genetics. So the issue turns out, now that we have these cheap sequencers and we can get a whole human genome for $1,000, the problem is, what do you do with that information? How do you make any sense of it? And that we're still working on. And in fact, I love this first quote. He's now the past president of the American College of Medical Genetics, Bruce Korpf, who said, we have a $1,000 genome, but actually understanding what we get out of that $1,000 genome costs $1 million. And so that we can't afford for all those genomes. And my buddy Russ Altman, who's at Stanford and one of the people who started computational biology and bioinformatics with me 30 years ago, noticed that it's not the price of computing, it's not that we need bigger computers or cheaper sequencers or stuff like that. The real problem is we don't understand what it means very well yet. And that's, it makes the field exciting and fun, but it also means that it's hard to explain when somebody gets a genotype or something like that, what significance it has. And that's where you guys come in. So I want to talk to you about some of the intuitions you have about genes and how they're wrong. So, and then not just yours. I don't mean to blame you guys, okay? So one of the things that you see in the newspaper all the time is scientists discovered the gene for whatever it is, you know, dancing or something. And that's always a lie, okay? There is no the gene for anything, okay? Genes work in big groups. So when you say scientists discovered the gene, they're wrong, okay? Scientists discovered a gene for that. And this chart, which I love because there is almost $1 trillion of research in there. Each one of those each one of the dots is a genome-wide association study. They cost more than $1 million each usually. There's about 500 dots in there. And let's see if I can explain what this graph means. So this, the odds ratio here, the vertical part, means if you have the particular variant, what are the odds that you have the phenotype, the disease usually? Okay, and notice that most of these things are hugging this line right around here that says if you have the risk allele, if you have the bad version of the gene, then you have a 10% increased chance of having the disease, a 20% increased chance of having the disease. It is not something that's fixed, all right? You didn't, oh, I got the bad disease gene, I'm going to get the disease. Not the way it works. Okay, there are a few that have very high odds ratio. So my favorite one is this one, MC1R. This is the only normal human phenotype that is inherited Mendelianly. All right, does that make sense, right? You know, you get one from mom and one from dad, and this is a recessive, so it means you got to have it from both sides, and there's carriers and all that. And what it is flaming red hair. Okay, blue eyes, by the way, is not one of those. Sometimes people think that blue eyes is Mendelian and inherited. It's not. It's more complex. Eye color is more complex. Lots of genes involved in eye color. But flaming red hair, there is one. By the way, and if you have the risk allele for flaming red hair, you have a more than 10-fold increased risk of having red hair. Do note, by the way, that this axis, the horizontal one here, is the reported risk allele frequency. That means how many people who have the disease have that particular gene, that risk allele, that variant of that gene that we found. And notice that of the people with flaming red hair, only about 10% of them have MC1R. All you other redheads, sorry, more complicated than that. All right? So again, most of these most of this is under 50%. So if you have the disease, do you have this risk allele? Maybe half the time for some of these. So genes work together in complicated ways. There is no the gene for X. All right? And by the way, let me dispel another common myth about genes. It's if you have the bad allele, if you have the disease allele, there's nothing we can do about it. Or maybe there's a drug, right? How many think, you know, oh, I got the diabetes gene, I'm going to get diabetes, maybe there's a drug, right? Not so. My favorite diabetes gene, we'll talk about it a little bit, has a terrible name. It's called TCF7L2. But TCF7L2 is really interesting. If you have the risk allele, you have a pretty dramatically increased risk of getting diabetes. So more than twice as big as people without that allele. And furthermore, if you're at risk for getting diabetes, if you have high blood sugar, you're pre-diabetic, we tend to give you a drug called metformin. And it's very widely taken. There's probably some of you in here who are taking metformin. The issue is if you have TCF7L2, if you have the risk allele, the bad one, metformin doesn't help you. And that's kind of a shame, right? Because we don't really have very much else to do for people who are pre-diabetic. On the other hand, if you have TCF7L2 risk allele, your environment is more important than other people's, okay? If you eat right and exercise, you are much less likely to get diabetes than other people in the same kind of blood sugar level, okay? So this is a gene that makes you susceptible to the disease, but makes your environment and your behavior more important than other people's, okay? Did you know there were genes that did that? Probably not. Okay. So I'm going to give you some of the basics and try and walk you through what kind of information we use when we're trying to understand what genes do. What are the resources out there? How are we going to do that, that analysis, that million-dollar understanding? So I want to tell you a little bit about the kinds of explanations biologists use with genes and where to find that sort of information you can use to put those kinds of explanations together, and how we can take pieces from multiple parts and different parts of the story and put them all together into a coherent mechanism is what we biologists call what they're looking for. How does it all fit together? How do they work together? And so I want to start off by pointing you to the good, reliable information sources. And so Ontology comes from the US National Library of Medicine. It's part of NIH. Ontology has these gene summaries. It puts together information from multiple different sources all in one big, long page, which I will show you in just a second and walk through it. The Europeans have a similar kind of thing called EBI gene summaries. EBI is the European Bioinformatics Institute. It's similar information. There are slight differences around the edges about what's included. It's presented in a somewhat different way. People tend to have favorites. I'm going to show you the US one just because we're here. There is a thing called a genome browser, which is a different way of looking at this sort of information. So genomes are linear, mostly. So DNA is stretched out in one long sequence. And so we can use that linearity to organize information about what's in it. And so we'll look at the genome browser from UCSC. And then, as I told you, genes come together in groups and do their thing together. And so I point to a couple of important databases about the groupings. So one is protein interactions. So you all know that genes code for proteins, right? Yes? Okay, good. They also code for other things, by the way. They code for RNAs that are functional as well. But proteins is mostly what we look at first. And proteins interact with each other. They're sticky. They have lock and key kinds of effects. And so one of the things that we have databases on that's useful for trying to understand what other genes might be relevant to the one that you're focused on at the moment is do the proteins they code for interact. And so INTACT is a nice database about protein-protein interactions. There are others as well. But with the bigger collection of genes that all work together on something are called pathways. So that's the one gene product will do something, and then let's take a classic metabolic pathway, digesting sugar. Okay, when you eat sugar, when you go have the lovely snacks that will be provided for you shortly, I am sure. Librarians always have good snacks. I'm not sure why that is, but it seems to be true. When you go have that good snack, the sugar molecules in there will be broken down and turned into something called adenosine triphosphate, which is kind of the AAA battery of your body. It's where we store energy. And the process of getting from sugar to ATP involves a whole bunch of chemical reactions. And that collection of chemical reactions are all catalyzed by proteins. And so the collection of proteins that catalyze the collection of reactions that get you from sugar to ATP is a pathway. All right, there's about 20 or so, depends on exactly how you want to count. Where we draw the lines around the borders of the pathways is a little bit arbitrary. But that collection of 20 or so genes that does something together as a unit is called a pathway. And reactome.org, that's the next one up there, is a nice source of information about metabolic pathways. And then I was told I should talk about pharmacology, so I'm going to include a kind of specialized database called PharmGKB. And PharmGKB is the pharmacogenomic knowledge base. It is a really well-curated database of polymorphisms, that is differences between people, in human genes that play a role in drug therapy, one variety or another, in medicine. And so PharmGKB is going to have that information about TCF7L2 or warfarin or Plavix, So it has, and it's very well curated. It's intended to be directly usable by docs who don't understand any of this, by the way. Doctors are not trained in genetics, okay? They get a little tiny bit. If they do pediatrics, they know about inborn errors of metabolism and the kinds of things that babies are born with, but your average GP knows nothing about genetics, okay? So you can help them. Let's pick one and do a little demo here. See if I can get this full screen. Yes. Okay. So this is entree gene. Let's try TCF7L2. And we get a bunch of TCF7L2s. So one of the things to know about genes is they're terribly named. Biologists did a really bad job of naming, not just because they're not memorable. TCF7L2 is not the worst of them. There's a gene called the, by the way. That was a really bad idea. There's another one called 2. The Drosophila, the fruit fly geneticists, they're the ones who really gave them bad names. Some of them are cute, but mostly it makes it very hard to search for them in the literature, which I care about. I do text mining stuff. But well, a lot of genes have the same name, but they're just in different organisms. So this is the human one. There's a TCF7L2 in fruit fly. There's one in mice. They all have the same name. So you have to say human TCF7L2. And they have lots of, TCF7L2 actually stands for transcription factor 7 like 2, which is a told biologist name. So we don't know what they are, so we have a whole bunch of them. They show up. It kind of looks like a transcription factor. That's a gene whose product interacts with DNA to turn on and off other genes. They're very important. Question?
How are the same gene different in human as opposed to mice?
Okay. So they have the same name because they're homologs. They arose from some common ancestor. So they're very similar to each other. They're going to have very similar sequences to each other. And we have it, and mice have it, and fruit flies have it because some ancient relative of all of us, we inherited it from all of them. Okay, so that's why they tend to get the same name. They're kind of the same gene, kind of. We call them homologs. And mostly homologs do the same thing. They have the same function. Not always. Sometimes genes diverge in their function. Then we call them paralogs, but I won't get into that today. Anyway, so this is what entree gene record looks like. It's so big, so big that there's a table of contents for the page. And a bunch of related information that isn't even on this page. But that's okay. So we start up at the top. The summary is a good place to look. It tells you a sentence or two about what's important about this gene. And it says things like, it's implicated in blood glucose homeostasis. That's why it's important in diabetes. Genetic variants of this gene are associated with increased risk of type 2 diabetes. And there's a widely used alternative name. Not only do genes have bad names, but they have several bad names. And this is where it came from. It's human. There's a bunch of related ones. HGNC is a nomenclature committee that tries to straighten out the bad names. Nobody uses them, but they do have good names, sort of. This is where it lives in the chromosomes, in the genomic context. So it's on chromosome 10. We all have 23 chromosomes, 22 numbered ones, and one sex chromosome given a letter. And so this is where it lives on chromosome 10. This is a particular position on the Q arm. You can kind of see some of the other genes that are around it. That's what these little gray areas are. Let's see, what else do I want to tell you about it? This is a bunch of alignments of specific sequences to the genome. This is a little bit of a genome browser here. So you can see, for example, genes have introns and exons. Okay, the exons are the part that will code for the protein, and the introns are spliced out of them. They have other signals in them sometimes. But as you can see, looking at this, go away, pop up, the dark bars here and here and here and here and here are the exons. Most of this is intron, not going to be turned into protein. You can see where it is. This is position 114,700 on the genome up to 114,009 something. Anyway, oh, I'm sorry, these are K. That's million, 114,700,000. Positions you can see down here, these are SNPs. SNP stands for single nucleotide polymorphism. It's a terrible name, means something that's different between me and you. Okay, or at least there's a potential difference. We've observed differences in human beings at those particular places. These are the variants, the differences we've observed in this gene that have been talked about somewhere in some paper. And the red ones are ones that have some negative thing associated with them. They're serious. That is, they usually mean protein coding differences. So that if you have that variation, your protein is going to be different than other people's. And so there are several of them in this gene. That's how that gets associated with diabetes. Okay. It has pointers to the sort of main articles about the gene. This is useful to people who like to read, like you. So it's going to say things like associations between TCF7L2 polymorphisms and risk of type 2 diabetes in the Chinese population. That's an important paper about TCF7L2. You could click on this and see all 450 papers about TCF7L2. The human one, there's others about others. And here's citations for homologs. So those are the ones that appear in other organisms. The fruit fly TCF7L2, the mouse TCF7L2. If I click on that, you'll see a lot of citations. These are gene riffs. I'm particularly fond of these. These are single sentence, usually taken from the title or the abstract of an article about it, that says something important about the gene. So for example, there's a significant association between TCF7L2 and risk of breast cancer regardless of ethnicity and or Native American ancestry. So it's not just about diabetes. Genes tend to, like I said, they have multiple functions. They show up in multiple places. So this one looks like it may play a role in breast cancer as well. A bunch of other things that you see down here. A strong correlation to variants of type 2 diabetes in Tunisians, in case you find yourself dealing with Tunisians. And I'm going to skip some of these other things. There's lots of interesting things in here. It plays a role in HIV. It interacts with an HIV protein. And here are a bunch of other protein interactions. So this is the protein, and it interacts with this other protein, and this is the evidence for it, and some little comments about what's going on, or what the evidence was for it, lots of those. Here are all the pathways that it plays a role in. Should take a look at one of these pathways. Let me see if I can show you what the pathway looks like. Diagram not available. Great, picked the wrong one. Back. Wnt signaling conserved. That should have a pathway. Yes, okay. So here is the Wnt signaling pathway. Can I make it a little bigger? No. All right. Yes, here we go. Wnt signaling pathway. Each one of these little boxes is a gene or gene product. This is outside of the cell, so Wnt is a receptor. It gets a message. It clicks in here. It does something to say, hey, I've been touched. I'm hooked up to my ligand. That changes this, communicates the message the inside of the cell, which is what's going on over here. There's a bunch of secondary messengers that change as a result of that message being communicated, and ultimately it turns on or off a bunch of other genes. This is a very important pathway in development. So this is all kinds of things that get turned on and off when this signaling pathway gets activated. Things like focal adhesion. Do cells stick to each other properly? And other kinds of signaling. The MAP kinase signaling is about whether cells should multiply and grow or should shrivel up and commit suicide. And so the balance in there says important things about both normal development and cancer. All right, let's get back out of this. Don't pick away. Sorry, this is not my computer, so I'm help. If I push the help button, it's not going to work. There we go. Okay. So now can we get back to, yes, okay. So one other thing I want to tell you about is this thing called the gene ontology. See gene ontology here? Ontologies are, it's a philosophic term. It means the study of what exists. In the modern world, it's kind of a special way of putting together controlled vocabulary. So it's kind of a list of formal terms that describe things. And here we're describing functions of genes. So we're looking at the function of TCF7L2 here, and so it binds to DNA, it binds to other proteins, it binds to nuclear hormone receptors. It does all that sort of stuff, and these are all the kinds of evidence we have for it, and when it says PubMed, that'll give you an article that has evidence that it has that function. The gene ontology talks about the functions of genes. It also talks about biological processes that they're involved in. So catenin import into the nucleus, for example, or embryonic hindgut morphogenesis, the building of your gut as you develop as a fetus. And it also says where it shows up. So where is it found? All right, I think I'm going to skip all the rest of this stuff. These are specific versions of the gene that have been reviewed by people, and where you can find it, and related sequences and alternative splices, and links out. Epigenomics is epigenetics, and therefore epigenomics, is about the kinds of modifications that happen to genes that don't change their sequence. So we can do things to turn genes on and off, some of which are heritable. If your mom turned off some genes using this epigenetic stuff, making attachments of methyl groups to DNA and stuff like that, you can inherit that as well. It changes over time, so if you're exposed to certain pollutants or if you develop in certain ways, you can change your own epigenetics as well. And we're just beginning to understand this stuff. All right, so I hope I didn't scare you with all of that, but that's kind of the information that's available and that you should help your customers go find. All right, come on, get me out of here. Good. I want to go back to the...
Are the 3D structures very clear?
Ah, yes, there are 3D structures here. This one, actually, I don't think has a solved structure, but usually you would find structures pointed to here, and there's a wonderful structure databases available. The main one is called the Protein Data Bank, PDB. We could, if you, PDB, protein, ought to be enough to, yep, there it is. So there's the PDB, and we get these beautiful, one of the things I love if you want to learn about protein structures is PDB does molecule of the month, and say pick interesting molecules, and they show you the cool structures, and you get these things. These are beautiful watercolors of molecules that show their structure, and you can actually buy these. These are great. Anyway, and it sort of describes in a nice, clear way what they mean, gives you, these are computer-generated structures that you can actually, I probably shouldn't do this. Anyway, they're 3D ones you can rotate. Yeah, I knew I shouldn't have done that. Oh, it's kind of working. Hey, there we go. All right. 3D structures. Okay, now, can I? Yes. Ha! Oh, I'm so happy. How am I doing on time? I have 15 more minutes, is that right?
Yes.
Okay, so I'm going to give you a quick look at PharmGKB. Thought I said yes. So PharmGKB is about the pharmacogenomic applications. These are things that play a role in, oh look, we got warfarin dosing, that play a role in medicine, mostly in the prescription of drugs. And so they do some nice work. They have these very important pharmacogenomic gene summaries, so you can go through the kind of main genes. This is only going to last for so long until we know a gazillion of them. But right now, we don't know that many genes that play a really important role in pharmacotherapy. So we can have little write-ups on each one of them. These are dosing guidelines based on a bunch of the genetic variants that play a role in prescribing drugs are ones that are involved in drug metabolism, the ones that make you a fast metabolizer or a slow metabolizer, and those tend to affect many drugs. So if you have one of those variants, it's not just one drug it affects the metabolism of, but many drugs. So we tend to write up summaries about those variants. They're called cytochrome P450s, helpfully abbreviated CYP. Sure, that makes sense. And they come in a variety. They're named with 2C, whatever. So anyway, these CYP2C19 and CYP2D6 are some famous cytochrome P450s. And so we can go pick some of these, pick a clinically relevant summary. I'm just going to take the first one. This is methotrexate. This is a cancer chemotherapeutic drug that turns out to have some other interesting applications as well. This RS number here names a particular polymorphism. That's a SNP ID. So it says this is a variant where you have a different gene here. It's a toxicity or an adverse drug reaction kind of evidence. And we get dosing guidelines. So, whoops, no dosing guidelines for this drug. Anyway, you can see all the things. There's the research about it. There's an overview. Some properties about the drug, mechanism of action, the pathways that it plays a role in, publications. So I'm running low on time, so I'm going to let you explore this, but it's a really wonderful resource, especially for the medical applications of genetics. let's see if I can get back to where it was. There we go. How about that? All right. So I just showed you 2 databases. There are 1,500 or so peer-reviewed databases that are kind of oriented around genes. Nucleic Acids Research did a really interesting thing. That's a journal, Nucleic Acids Research. Starting about 10 years ago, they started peer-reviewing databases. So as you probably know, scientists get credit for their work by getting peer-reviewed journal articles. And so the scientists who put a huge amount of work into building these databases got no credit for it. And so Nucleic Acids Research did something to try and get them credit. So there's an annual database issue. It is the first issue of the year, January 1st. So while you're recovering from New Year's Eve and watching bowl games, you can check out the new edition of the Nucleic Acid Research Gene Database Collection. This year there were 1,560 of these databases that had passed peer review. They're specialized in all kinds of ways. But it's a great resource. It means that the databases, some scientific expert has looked at these databases, or usually multiple experts, and blessed them. So it's a nice, reliable source of information about all kinds of, if you care about amino acid, tRNA synthetase genes, there's a database of those. And it's much more specialized information than is in these general databases that I showed you. It is a little overwhelming. 1,500 databases is a little much, but nevertheless, they're nicely indexed, and you can probably find the one you need. Yes, sir?
So this issue, that once it's issued, it's kind of like a consumer reports of these databases, so...
Yeah, a little bit, except for they'd leave out all the bad ones. So only the good ones are in here.
Okay. Who uses that? I mean, who's the user, and where?
Robin, me, people who need specialized information. I find a collaborator in neuroscience. I'm not usually a neuroscientist, but I've done a little bit of neuroscience work. A collaborator comes in and say, I'm really interested in understanding variants in the structure of dopamine receptors. And it turns out there's a dopamine receptor database that has all kinds of information about how variations in dopamine receptors affect behavior in mice and rats and people. And I can use that to try and explain something about what I'm seeing when I see some variation in dopamine receptors. And I probably wouldn't get that directly out of the big database because, first of all, it doesn't mix together human, mouse, and rat. And I'd have to go follow the links to the articles to kind of read that stuff. And so those databases centralize useful information for very specific consumers. If I have a new database that I put up, I think I have a better way of searching, a better way of visualizing, something new and interesting, I will send it to Nucleic Acids Research and hope that they like it. Okay? All right. I'm going to race through this because I've mostly done the rest of the things I wanted to say. So I talked a little bit about the functions of genes. All of this ultimately relates back to the things that organisms do, which is turn stuff around them into copies of themselves or into themselves, right? So that sugary snack is really about making more of you. And then, of course, we have the imperative that all organisms pursue, and that's reproducing. And so in order to do that, they have to metabolize all the stuff that they suck in and turn it into things that are themselves. They have to make their bits and pieces. They have to make DNA. They have to make proteins. They have to make fats. By the way, lipids are vastly underrated. People think of fat as one thing. It's not. There are as many different fats as there are genes and proteins. And they play very different roles in signaling and the like. And it's very important. So don't be anti-fat. We have to sense the environment around us. We have to hear and understand and see and taste and feel and all that kind of stuff in order to grow and reproduce properly, not to mention take care of our offspring. And we have to maintain what biologists call homeostasis, that is, stay within the range that we can survive. Okay, so I can't have too much or too little water. That's why you're all hydrating before your hike. And I need the right amount of food, and I need the right temperatures and all that kind of stuff. So these are the functions we're going to see, and these are the ones that are going to explain when you see some set of genes that do something, ultimately understanding the mechanism of what those genes are doing is going to be related back to this stuff, sometimes very remotely. Sometimes the path is a little long, but that's, I'm going to skip this, I'm going to skip this. I do want to talk very briefly about transcription factors. So we tend, another one of those mistakes about thinking about genes is we think a gene it does one thing and kind of in isolation. That's not only not true because they all work with partners, but it's true because genes affect lots of other genes. And the most important of those, the ones that have the biggest effects, are these things called transcription factors. And transcription factors are genes whose protein products bind to DNA, interact with DNA, and in multiple places all along the genome. And when they do that, sometimes in partners with others, they cause the gene near there to be made into protein. Okay, so they cause the transcription of that gene. And they can cause the transcription of many genes. So one of these proteins can change the way lots and lots of different proteins are being made, how much is being made. So often DNA is thought of, or the genome is thought of as a blueprint. These are the ones that are kind of given the construction orders to say, okay, make lots of copies of this one and fewer copies of that one, and we need to bring them all over here, that kind of stuff. Okay, so yes?
So statin drugs then, would all the just different statin drugs, do they affect the cholesterol, the lipids differently on different people?
Yeah, sort of. Not related to transcription factors directly. That's okay. So statin drugs interfere with mostly the metabolism of the production or consumption of cholesterol, and some of them interfere with transport. So cholesterol is good for your cells when it's inside doing what it's supposed to be doing, and bad when there's too much of it floating around in your blood and sticking to the sides of your veins. So the cholesterol drugs work in different mechanisms. Some of them inhibit the production, so your body produces less. Some of them change the way transport works, so your cells are more effective at sucking it in. And most of the cells that suck it in are in your liver, by the way, also the ones that produce it. All right, so let me just show you a little graph that takes one typical transcription factor. And so these purplish bars are without the transcription factor, and these reddish bars are with it. And each bar is another gene, and you can see that it changes the production of many of these genes, especially over time. So this is in embryonic stem cells, and this is in 8-day-old embryos. And so this is one of those early transcription factors that influences development. BMP actually stands for bone morphogenic proteins. So I mentioned a little bit about the gene ontology. The gene ontology, I think, is a really important resource in describing the functions and processes and locations. There's some other ontologies that have been developed that are sort of related to it. And it has this sort of hierarchical structure. So down here is transcription factor binding. That's a function, something a protein can do. It can bind a transcription factor. But not only is this a controlled vocabulary, it's a hierarchical controlled vocabulary. So transcription factor binding is a kind of transcription factor binding activity. It's one part of the activity. It has to not just bind, but it has to actually recruit the other protein that turns on the transcription. It's a kind of protein binding, which is a kind of binding, which is a kind of molecular function. So these functions and processes and the like end up in combinations that sort of define what it is that they're doing. And so we could say that it regulates something, or it's part of something, or it's a kind of something. And so the ontology reflects that. Yes, ma'am?
So you have the hierarchical structure there, and all these auxiliary proteins that are coming in, are those sites then for drug interaction and therapeutic work?
Sometimes, right? So drug discovery is a complicated business, but the simple version of drug discovery is we find some naturally occurring, usually protein, that affects what we want to affect. Usually we look for ones that are those receptors. See, I shouldn't have skipped this. So the ones on this is a cell membrane here, and there's some receptor. This is a very commonly drugged kind of protein called a G protein linked receptor, or G protein coupled receptor. There's some kind of normal signaling molecule that activates that and causes some downstream activity to happen. So often our drugs target these receptors because it's much easier to get a drug in a reasonable concentration on the outside of a cell than it is on the inside of a cell. So we come up with one that gets in the way and prevents the signaling molecule from binding, or acts like the signaling molecule and turns it on even though the signaling molecule isn't around. And so that's a main mechanism of drug action. And an awful lot of drugs, by the way, target G protein coupled receptors. And so if we're trying to invent a new drug, we look to find the receptors that do the normal version of what we're trying to do, and then we try and come up with something that acts on them.
Thank you.
Sure. All right.
Mary, you have a little wiggle room.
Okay, no, I'm gonna quit. I'm getting tired of hearing myself talk. All right, so genes work together. We got to understand how they work together. So there's various ways to do that. One is called function enrichment. So I can take a whole bunch of genes that are all annotated to the gene ontology about things they do, and I can say, okay, let's look at all of the different pieces of the gene ontology and see which ones are lit up by my big pile of 100 different genes, and which ones would be lit up by a pile of 100 random genes, and what are the differences. So I will see places where my particular 100 genes light up the ontology in a way that a random 100 genes wouldn't, and then I can color code them. So this is a chunk of the ontology that you can't read, but that's okay. The point of this is, gee, my collection of genes is really lighting up these functions and kind of lighting up these functions and not so much lighting up those. Okay, so that's function enrichment, sometimes called gene set analysis. And that's an important way of understanding how a bunch of genes working together, what are they doing? How can we understand them as a whole? Here's another way to do it. We can take the pathways, and we can color code the pathways by which genes we're seeing. And so when you light up a pathway based on a whole bunch of genes, they all tend to congregate around this pathway and not so much that pathway. That's another way of understanding what they're doing together. That works great when it works, but our knowledge of the pathways that genes are in is pretty sparse. So maybe somewhere between 10% and 25% of genes participate in some known pathway. Other than that, we don't really know what they do. I'm showing you genes intentionally where we know a lot about them. It's often the case that some gene will pop up in some experiment and we know very little about it. There's still, you go to the same places and do the same things, you just get back fairly empty searches and not much is known. But that's why we do research, right? Try to figure that out. I love this because I'm interested in biological visualization, and this particular way of lighting up bits and pieces and redrawing bits and pieces of pathways won the Biological Visualization Conference Best Paper Award. It's something called En route, and there's a URL that will get you to the website for it. Okay, all right. This is where I said most of the time we don't really know, sadly. These are the kind of cartoon diagrams that biologists draw for each other to try and combine various functions together and communicate a story. Each one of these things is a gene. The box around the outside is a cell, so you can see these are the receptors. The receptors receive a bunch of different signals. Those signals are all integrated together, and ultimately this is this MAP kinase pathway I told you about a moment ago that controls whether or not the cell is going to multiply and grow or undergo something called apoptosis and commit suicide. Apoptosis is spelt like apoptosis, by the way, but it's a PT like the pterogram or something like that, so the second P is silent. Most people get that wrong. Sorry. All right. And I think this would be a good place to end up. There's lots of challenges to doing this, okay? This is not an easy task, providing the information. That's why it's $1 million interpretation for that genome. There's a lot of kinds of knowledge that have to be put together across lots of different application areas. So your biologist coming up to you and asking for help in understanding this collection of genes is going to be expert in some areas and know nothing about others. And the information resources that you have are going to cover all of these areas. So there's a ton of information that's very diverse and hard to integrate. A lot of biologists are not very trusting people. Docs likewise. They want to know what the evidence is for things, so you can't just give them the summary and say, okay, here's what the database says. They're going to want to know where the evidence was and what kind of experiment it was and how good were the controls and all that kind of stuff. So you have to be able to get access to those experimental justifications. I think I pointed out the gene ontology has an evidence code that says what kind of evidence it is and a PubMed link that gets you to the article that had the evidence. There's a lot of complexity in the way that genes can play a role in some disease or something, some other normal human phenotype. They can play a role by not being there. That's complicated. So when you search for things that are, okay, what are the genes that are involved in red hair? Sometimes one of the genes that's involved in, say, red hair is something that isn't expressed and isn't going to be listed in the ones that are involved. But if it were there, then you wouldn't have red hair. So there's logical complexity to how these things all fit together. Often the fitting together is of more interest than the individual pieces. So you may have heard the term systems biology. It's kind of a hot term these days. And what that means is we're going to look at all of the things together rather than going into sort of reductionistic detail about the explicit activity of each one of them. And that's been very, very useful in trying to understand biology better. Part of your job, as you well know, is to sort through the amazing amounts of information and try and hand people the stuff that's actually relevant to them. That's particularly important in this area. As I showed you, just looking at one database took me 5 minutes to just scan through and tell you what the things were, let alone look at all of those things. So it's really important to be able to understand the user's context and what they need and be able to provide them the information that's relevant to them now and pointers to other stuff that they might want to look at elsewhere. And as you probably know, the biomedical literature is growing at leaps and bounds. It's also growing exponentially. Last year, for the first time, we hit 1 million new articles in PubMed for just 2012. Okay, if you do the math, that's about 2,500 a day. And one of the interesting things about the modern world of molecular biology is we used to be able to specialize and just read my journal and stay reading my field, but the damn genes don't respect our disciplinary boundaries. And it turns out that you may be studying this gene in the context of diabetes and development, but it turns out some really important work studying that gene was done in neuroscience or cancer or in kidney failure. And not only do you have to find those articles, but you have to make sense of them. They use different vocabulary. They assume different background. It's really hard to integrate all the information that's out there about particular genes because of this sort of contextual problem. And so text mining approaches and other sorts of ways of trying to automate taking some of this information out of the literature are particularly important in biology. All right, I've gone 5 minutes over. I'm sorry about that. I hope this was useful to you, and I look forward to hearing what Robin has to say.
So now we're getting ready to have Robin, Dr. Dowell, speak to us. Robin is an assistant professor in the Molecular, Cellular, and Developmental Biology Department and a member of the BioFrontiers Institute. She is a Betcher investigator and Sloan Fellow. Her primary expertise is in computational biology, specifically using machine learning techniques to better understand genomics. Her laboratory studies both the evolution of transcriptional regulation and how variations within a species contribute to phenotypic differences, a key aspect of personalized medicine. Prior to joining the faculty at University of Colorado, she worked as a postdoc at MIT and completed her graduate work at Washington University in St. Louis. Please welcome Dr. Robin Dowell.
All right, so I want to thank you guys for this invitation. My grandmother was actually a county librarian. And my parents attribute most of my interest in academics and science to her and her reading to me. And one of the things I regret very deeply is that while she was alive, I could never explain to her what I was interested in or did. I was a college student studying computer engineering and taking a ton of genetics classes on the side. In the end, I ended up actually as a double degree. And I would go visit my grandmother, and she'd say, so what are you doing, and what are you going to do with your life? And I would launch into these stories about medicine and genes and programming and computers and databases, and her eyes would glass over. And she'd look at my roommate and say, well, what are you studying, and what are you going to do with your life? And my roommate, God bless her soul, she was studying environmental science, and she would look at my grandmother and say, I make the water you drink safe. So today, I can't get to one sentence, still, more than a decade later. But hopefully I can get to a description that librarians might appreciate. So today I want to talk about your genome, and Larry gave a really great introduction to sort of how complex this is and how much information there is out there. But I taught for the first time, so I'm fairly new here, been here 4 years, but one of those years on maternity leave, so I'm still a very junior investigator. I taught for the first time one of these undergraduate genetics classes that has 350 students in it. And in the process of doing the lectures for that class, I came across a statistic that scared me. And that was in 2010, at one of the large AMA conferences, lots and lots of doctors, they were polled and asked, where does your genome live? And only about 50% of them got it right. So as librarians, let's start with where does your genome live? Okay? So every cell of your body, I don't know if this pointer is going to work for me. Hey, look at that. I've never been double mic'd before. Every cell in your body contains a nucleus, and inside that nucleus are the chromosomes that Larry mentioned. And you've got 23 sets of these. And they're basically composed of a series of proteins that pack them up and read from them. But then there's this long stretch of nucleotides that is the DNA, which we've been so, intimate with these last several years. And when you're sequencing a person's genome, you're taking cells from their body, grinding them up, pulling out that DNA. I'm going to keep pulling this down until hopefully we can get away from the feedback. Sending it to a machine like this one, which looks a little bit like something you'd see in a Star Trek movie with, a blue light that just. It actually reminds me of Knight Rider from my childhood. But, and you throw the biological sample into this thing, and you wait a couple of days, and what you get back looks a little bit like this, or at least the background there. Lots and lots of A, T, C's, and G's. It's a book. We just can't really read it in enough detail yet. We're sort of like handing the Encyclopedia Britannica to my son, who's about to start the first grade, and saying, read this and understand it. He gets a lot of the words, but he can't get all of the meaning or the nuance. But the cell can take this book and read it and create sort of the vast diversity you see in this room. So the genome is thought to be the book of life. So maybe you had a little different perspective on what you were made of. But as Larry mentioned, acquiring your genomic sequence is getting cheap. Now, this is a slightly different version of the same graph. I thought very seriously about stealing Larry's slide when he said, here's what I'm going to talk about. And then I thought, no. I'll stick with this. So in 2001, the human genome was published. So what is the human genome, the one that's in the database? Well, it turns out it's not actually any one person on the planet. It's a haploid genome, meaning there's only one copy of every chromosome. You have two. It's a mosaic of many individuals. So it's not really representative of a human. It's a reference. It's sort of like having a Rand McNally map. So I don't know if you guys are familiar with it, but Rand McNally puts intentional errors into their maps so that they can detect plagiarism. So they add cities, put roads together at the wrong locations. I'm not saying that the human genome's wrong, but it's a consensus bit of information, which means medically, perhaps it's not really the best reference for an individual. So if I go to the hospital, and it's now somewhere between $1,000 and $3,000 to sequence a human genome. If I go to the hospital, if it were medically useful, they could, for that amount of money, send my cells off and hand me a DVD with my genome on it. And hopefully somebody, and Larry mentioned that the interpretation of this is actually what's hard and expensive, somebody could hopefully look at that genome and use it in a medically relevant fashion. That's the goal where we want to get to with what we call personalized medicine. But we're very far from there. So how do you get to personalized medicine? So if I were to draw a path, one of my Rand McNally maps, we got to start with gathering that raw data. And this is where we've been for the last better part of a decade or more, to get at complete genomes. So the first genome, this mosaic, was published in 2001, but the first individual's genome was published in 2007 or 8, I think it was. And now there's a few hundred of them floating around that have individual papers on them, and there are projects that are collecting thousands of individuals. There's a Thousands Genomes project, which, despite the name of saying 1,000 genomes, is trying to sequence about 3,000 people. So we're not good at math. But we're sort of stuck here in this green to starting to turn to blue region, as Larry mentioned, where we're trying to get at interpreting all of this information. And so, this is really where I spend most of my life, is on that puzzle right there. So Larry likes scientific art, so I found the prettiest picture of the central dogma I could find. Okay, so here is a cell. There's the nucleus. Coming out of that nucleus is the DNA. I've got machinery that's converting that into RNA, and it's converting another machine that's converting that into protein, which is folding up and making this machine. I like things a little simpler. This is the central dogma. This was postulated years ago that DNA is a book. It's read into RNA, which you can think of primarily as a messenger, which it's funny that I'm saying that because I've spent my entire research career studying RNA and trying to convince people it's not a messenger, not just a messenger. And those messengers are then converted into the proteins or the machines that make the body work. And an awful lot of what Larry was talking about was what we understand about the machines. Now, he used the word gene. And in my opinion, gene has got to be one of the most poorly defined words in the English language. A bunch of biologists could get into a room like this, listen to 20 talks, all of them they use the word gene, and they all mean actually different things when they personalize or internalize the meaning of the word gene. So for the sake of this talk, I'm going to use gene in the way that is usually implied by most biologists. And that's to mean these proteins are these machines. And Larry said that most genes, or most human diseases, by the way, are caused by multiple genes, and that's absolutely true. But there are many genes, and here's a list out of a standard genetics textbook, that when they malfunction can create disease. Marfan syndrome, for example. These are what you call the easy ones. If this disease is this gene is broken, this person is highly likely to get this disease, such as flaming red hair. I had never heard of that. But this is not the norm. This table is not that big. Now, these are only dominant genes, so you only need one mistake and one copy of these to get this disease. The norm is what Larry showed you. Things like heart disease, type 2 diabetes, which unfortunately I'm in that camp, that are caused very complex, caused by lots of different genes malfunctioning. So let's look, we're 12 years past the original Human Genome Project. What does a typical genome tell us? What do we know about a typical genome? Well, it's composed of proteins, which if you squint your eyes is this tiny little green wedge right there. Those are the machines, which means 98% of your book is not making the machines that make you function. What does it do? Unfortunately, probably about 50% of it is repeats, and there's huge controversies on whether repeats are important, because the other word we have trouble defining is important. Almost all genes have alternative splice forms, so yes, we have about 20 to 25,000 genes, but they can be read in different ways. You can read the sentences basically different, if you think about it, this is like being in those old English classes that I was always so bad in. Put the punctuation in the sentence. Well, if I move the comma, I can get the same sentence to mean something else, right? And isoforms are sort of like that. So they can read the same region of the genome in different ways with different inflection. But most of the genome, despite not making these machines, is still read, still transcribed is the way we would use this or describe it. So what does it do? Or does it do anything at all? And that's where I've sort of spent a huge amount of my time studying this RNA and this process of reading the DNA into the RNA. So recently there was a project called ENCODE. Maybe you saw some of the press about this. ENCODE claims 80% of the genome, even some of that repetitive stuff, is functional or important. But they didn't really define function or importance, so again, we're sort of back into this argument. But from the standpoint of this talk, and from the standpoint of you going to the hospital and getting your genome on a DVD and sliding it in your back pocket, what's different between you and the person sitting to your left? You're 99.9% identical. So you have roughly one difference in every 1,000 base pairs. Those differences can be a character changing. So I can, make the word tap into the word tat by changing the P to a T. They can be insertions or deletions. I can add whole words, sections, letters. They can be copy number variations. I can take a sentence and copy it many times. So many of the same sort of edits that you're used to when you think about text. But between any two people, 1 in 1,000 doesn't sound like a lot of variations, but when our genome's 3.2 gigabases, it ends up being about 3 million variants between any two people. And those variants obviously give rise to huge phenotypic differences. Can we leverage those medically? So I highly recommend everybody go read this article at National Geographic. So Lily Grossman, small child, when she was very, very young, I want to say 12 to 18 months, she started waking up in the middle of the night screaming in pain. At first it was once, maybe twice a night. Rapidly within a few months, she was waking up almost every hour, screaming in pain. I don't know how many of you have been up with a colically child, but this is an ultimate form of colic and sleep deprivation for her and her parents. She started having muscle wasting and basically could never sleep more than 40 minutes without being woken up in excruciating pain. And it was a systemic pain, so she couldn't even say where it hurt. So they took her to the hospital. As it turns out, many, many hospitals, had many, many tests, all of modern medicine. She was diagnosed with Duchenne's muscular dystrophy. That was wrong. She was diagnosed with a particular psychological disorder. That was wrong. She was diagnosed with 10 or 15 things, and everything was wrong when they then tried to treat it or they tried to do subsequent confirmation tests. So her parents sort of became their own best doctor, her own best doctor. And they started reading the literature and going to their local librarians and ended up discovering that on the West Coast, there were hospitals that were taking patients just like Lily and sequencing their genomes and trying to figure out what was wrong with them. So they picked up their entire family and moved to the West Coast and actually moved very close to the hospital. Met the doctors that were doing this. They decided Lily was a great candidate. They sequenced her genome. From the point in time she went in for the blood draw to have her genome sequenced to the point in time they had the full genome and knew what variant she had was about 3 to 4 months. At this point, she was 12, 13 years old. Still never in her life slept more than 40 minutes. Well, not since she was about 12 months old. So the question was, what was wrong with Lily Grossman? So 3 million variations between her and any other genome they looked. So how do you find in that 3 million what's causing that disease? Well, in her case, they sequenced one of her parents. Well, actually, both her parents. So you could tell in Lily's genome which mutations had arisen uniquely in her. And they went and they looked at those genes, the proteins. And there were 2 proteins with unique mutations in Lily's genome. Neither of those mutations had ever been seen in anyone ever before. One of those genes was involved in a muscular phenotype, and the other one was involved in pain sensory neuronal type stuff. The neuronal one, essentially completely unstudied. They barely knew it was neuronally associated. The musculature one had been studied for an entirely different reason. There happened to be a drug out that was used to treat patients that had a different problem with that gene. And so they decided, what do we got to lose? Let's give Lily the drug. So they gave Lily the drug, and 3 nights later, she slept for 18 hours. It's not a perfect story. She has since started to have wake-ups again. They're trying to muck with the drug. They're trying desperately to figure out what the other mutation does. It's clearly a complex story, but this is one of the cases where genomics has come through and explained something that we had no way of going at in any other fashion. So I mentioned we have 20,000 to 25,000 genes. It turns out that the worm, the nematode worm, has about 12,000 genes. So there's 2 ways to think about this. One is, we must be doing something really cool with that extra 10,000 genes. The other is, maybe we're missing something, okay? So what makes us different from a worm? So there's a guy, John Maddock, who's actually out of Australia, who for a number of years now has been looked upon by some as a complete and utter quack, because he's been saying it's got to be more than just the proteins. It's got to be more than just the proteins. And these are figures from some of Maddock's reviews. And one of the things that Maddock noticed was the more complicated the organism, the more non-coding genome it had. And I'm not talking the repetitive parts, I'm talking the non-coding parts that aren't the proteins and aren't the repeats. Larry mentioned these briefly, and what Maddock originally noticed was the introns, the parts of the genes that are transcribed and then spliced out and thrown away. Seems kind of wasteful. Why are those getting bigger as we get more complex? So the worm, which is here, C. elegans, has 24% of its genome as coding. We have 2%. The worm has about 30% of its genome transcribed producing RNA that does not appear to make a protein. We have 47. And what we now know from projects like ENCODE is that if I look along the x-axis here, sorry, let's go with the y-axis first. Nope, I'm right, the x. I can't read these on my screen, so I'm having to remember. The x-axis, as I move to the right, is how often I see that molecule in the cell, how often it's transcribed, how often is it read out of the genome. And what you immediately should see is that the protein coding stuff is read much more often than the non-coding and many of these small intergenic RNAs that we're now currently discovering. Furthermore, if you look at the other axis, the y-axis, it tells you how much of this stuff that's read just stays and hangs out in the nucleus versus goes out into the rest of the cell. And most of this RNA stuff appears to hang out in the nucleus. Doesn't appear to leave it, which has led some people to say it's junk. But something has to tell that cell when to turn on what. Something has to regulate how to read that genome. If you think of this as the genome as being a score, you still need a conductor to work the orchestra. So maybe these guys are involved in regulation of that genome. So here's another metaphor. If my DNA is the planet, and I start taking photographs with my molecular techniques of the entire planet, the things that are expressed highly are like the bright lights of the cities. And if you tried to understand the planet by looking at these photographs, you would think everything happens in New York and Chicago and Houston and Los Angeles. But in fact, most of the food that we consume is grown in these dark areas. So just because the bright lights are easier to study, they've gotten most of the attention, but the dark areas are not necessarily unimportant to how that planet functions or how your genome functions. So one other interesting result out of ENCODE that sort of helps us to further fund our research because it convinces people what we're doing is not quackery, is that depending on how you think about these variants that Larry described that cause a particular disease, type 2 diabetes, if I now associate back to the genome, these locations in the genome appear to, when it's changed, associate with this disease. What's actually there? What can we go look and find? And what we find is only a small fraction, even in sort of the loosest criteria, actually correspond to the protein coding. The rest are transcribed or in regions known to be involved in regulating when something is read or transcribed. So maybe what makes us human and not a mouse or a worm is not the proteins, but how we use them. We have opposable thumbs. We can make tools and use them. Okay? So that brings me to, what does my lab do? So we study individual variation within a population. What's the differences in this within this room? And how do those variants contribute to how differences in that DNA being read, how that score is orchestrated? If I give the same score to different conductors, I will get slight variations on the music. And you see that in identical twins. Okay? So we tend to look at genomes of lots of individuals within the same species. And the idea is what variations within, say, this person, Chamberlain, makes him look somewhat distinctly different from this guy, or in yeast, or in mice. And what we bank on is that evolution is going to help us. Okay? Just as an aside, my husband is an children's book illustrator, and so the one on the left is actually his. And so what we're banking on is that if it's important, most of the time it's not going to change. So we want to look at the variations you do see between two individuals and distinguish which ones of them are random noise and which ones are contributing to phenotype. So here's the first, what I would call scary slide. So in detail, this is what we do. Okay? We do, some amount of experiments, in this case on yeast. We throw them to that Borg. It gives us back a bunch of data we have to store in some giant machine that could probably heat this whole building all winter. And then we have to do a whole bunch of that really expensive analysis that Larry was talking about. So I want to give you 2 vignettes on 2 projects we're working on in the lab right now. And I'm sort of going out on the limb and actually talking about stuff we haven't really published yet, because I think it's just too cool not to share. And the first has to do with alcohol tolerance. Okay? And so as it turns out, a lot of how sensitive you are to an intoxicating dose of alcohol is determined by your genes. And it has been shown in mouse models and subsequent human studies that your predisposition to drink, particularly after your first exposure, is largely genetic. And so there's been a mouse model developed where mouse were bred, and I'll go into this in a little bit more detail in just a minute, where we use something called a loss of writing reflex. So we give mice an intoxicating dose of alcohol, massive dose of alcohol, and literally they pass out. Okay? And then we measure how long it takes for them to get back on their feet. And what we know is that the longer they stay passed out, the less likely in subsequent exposure to alcohol they are to drink. So if the next day we put 2 tubes of water or 2 tubes of liquid, 1 water and 1 alcohol, they'll avoid the alcohol like a poison if they're a very long sleep. Sorry, we use the phrase sleep. Long sleep mouse. If they pass out and they wake up very quickly and you put the 2 containers of liquid, they'll basically keep themselves buzzed forever. And we've been able to show, or I shouldn't say we, this is actually all my collaborators work down at the medical school, Richard Radcliffe, has been able to show that this actually is strongly similar to humans' propensity to, after going out on a night drinking, want to go out again the night after and do the same. And it's genetic based on twin studies. So what Radcliffe has spent a huge amount of his research career on is developing a mouse model to study this. Because we really want to understand these phenotypes in humans, but humans have a little bit of a few problems from a scientific standpoint. They don't like it when we tell them how to breed. They don't like it when we ask them for a piece of their liver. They don't, we got to have some other way to go after this to really understand the molecular basis of these diseases. So what Radcliffe's mentor did, and actually Radcliffe came in on this project after it had gotten started, but he was heavily involved, was they took 8 strains of mice, laboratory strains and wild strains, and started breeding them and selecting within those breedings for strains that were either more sensitive to alcohol or less sensitive to alcohol, as defined by this loss of writing reflex assay. And what they ended up at the end were 2 strains of mice, one that they call the long sleep mouse and one that they call the short sleep mouse. And what's plotted there are how long they were asleep after they were given this intoxicating dose. And in black is whether or not on the day before they got a really light nip of water versus a light nip of alcohol. Because the question was maybe, if it's really just that first exposure to any alcohol, maybe we would desensitize them if we gave them a little nip that didn't make them sick. Turns out that's not true. So what we wanted to do, this is where Radcliffe has spent most of his career. And what he's been doing is doing these genome-wide association studies that Larry mentioned, looking for genes that are involved in alcohol sensitivity, alcohol tolerance, in these 2 strains of mice. And you get a plot like this. In mouse, there are only 18 chromosomes, and each one of these little marks with a name associated with it is a location where we have found an association with a particular alcohol-based phenotype. The one that you want to pay attention to is the LORE, the loss of writing reflex. I don't know why it's E and not R on this figure. And so as you can see, there are lots of genes or loci, locations, in the genome that seem to be associated with this loss of writing reflex. And when we look at the mouse genome, we see the same thing that ENCODE saw. Small percentage of these are associated near a protein coding gene. Some of them appear to be transcribed, and many of them appear to be in the desert, and it's really not clear what they're doing. So the first thing we wanted to do was to say, well, maybe in these 2 inbred mice, in those desert regions, there's been some variation that would help to explain what this does. And so we wanted to sequence those 2 long sleep and short sleep mice, the extremes. So we, again, got some mouse liver. I apologize. Some people get very upset about that, but we got some mouse liver, isolated the genomic DNA, and did library propagations and threw this at one of these board machines to get back sequencing. I'm only going to talk about the first initial sequencing data because all the stuff on the right here, I can honestly tell you we got at the end of May of this year. So I don't really know what's in there yet. But this first Illumina run on the short read library, short insert library, came back with 427 million base pairs of sequence for each of these 2 mice. So a mouse genome is about 2.8 gigabases. So this means every position in the mouse genome we had sequenced on average roughly 32 times. You have to have that kind of redundancy statistically sampling in order to have any chance of assembling this thing back out, because what I didn't tell you is that those sequencers, while they're really good at producing a ton of sequence, they do so in like 50 base pair chunks. So if you got a 3 billion base pair genome, it's sort of like me walking up to you with a puzzle that's 3 billion bases and saying, here, put this together, I'm not giving you a picture. So the way we do this is we sort of divide and conquer. We sequence it many, many times and look for places where they overlap and try to build this thing back up. And so we did that for these 2 strains, and then we compared it back to the mouse reference. And in the mouse, the reference is also a mosaic, but it's less of a mosaic because it all came from an inbred line. So it's a little bit more representative of an individual. And we found about 9 million places where one of the strains varied from the reference or the other strain. That's a lot of places to look for what's causing these alcohol phenotypes. So we started comparing it to variants that had been seen in other mice, asking whether or not our software was actually accurate, which I hate to admit how much time my lab spends on that. And it's not just my software, it's any publicly available resource tool. And what we ended up was with about 3.5 million SNPs of high quality or variation. I shouldn't call them SNPs, some of them were actually insertions and deletions, that looked like they're bona fide, they're real, they're not noise in the data, and they potentially could be of interest. And so now we went back to the genome again and said, what do those overlap? And just like ENCODE saw, a very small fraction, which are these little bits up here, overlapped protein coding. The majority of them were either intronic or between genes. So this doesn't help us that much with interpreting how are these guys more or less sensitive to alcohol. But we took these ones that are overlapping the genes and we said, do any of those guys look particularly promising? And there are a few genes that have been associated in these mice with the alcohol phenotypes, but they explain very little of the phenotype. And this plot kind of explains why. If we just randomly pick places, we get a plot similar to that pie graph I just showed you. But the likelihood of it associating with the phenotype, not so good. So this leaves us with 2 conclusions. One, an awful lot of the variation between this mice that's driving their alcohol sensitivity is not protein coding. We don't understand how or why yet. And 2, the protein components that are different don't explain very much of the phenotype. So the next step, and what we're currently working on, is we crossed, I shouldn't say we in this case, Richard long ago crossed these two long sleep and short sleep lice and built what's known as a recombinant inbred line. So you take your two inbred parents, you cross them to make F1 or the first generation. You start self-crossing those to make an F2, and you keep that process up until you end up with strains of mice that are essentially the two chromosomes they contain are identical to each other, but they show variation in which parts of their genomes came from those original two lines. We've got 75 of those. And what's great about that is now if a phenotype exists in a subset of that 75, 20 of the mice, that looks like mom, the original long sleep mouse, I can say what parts of their genome do they share with that long sleep mouse, because those have to be the places where that phenotype came from. Okay? So we can leverage that now to do transcription studies in these mice and ask, for all of these variants we see between the two original long sleep and short sleep mice, how are they actually affecting transcription, and does that relate back to the alcohol phenotype? As an aside, because you guys love information, this part of the project, we are doing RNA-seq in 52 mice in 3 replicates in 2 different conditions, saline versus alcohol, which means when the whole project is over, we will have, I really gotta move, we will have roughly 16 terabytes of raw, unprocessed, never before analyzed, haven't done any analysis, it's just coming off this machine data. And that's what we have to sift through to figure out how these variants contribute to alcoholism. So the other big project in the lab involves copy number variation. And you're all familiar with copy number variation, whether you realize it or not, the most famous case of this being Down syndrome. So a Down individual has 3 copies of chromosome 21, right here. And despite the fact that copy number variations in our genome are some of the earliest medically described conditions, we have no idea from a molecular biological standpoint how the cell reacts to having an single extra copy of one of your random chromosomes. So there's a whole chunk of my lab that's interested in aneuploidy issues. So it's not just relevant to individuals with Downs who are, rocking that extra chromosome, but it actually also happens to be really important agriculturally. Most crops are aneuploid. Happens to be important medically from the standpoint of most bacteria and fungi that become antibiotic resistant do so by becoming aneuploid first, and we're not really sure how that is related. And most cancer cells are aneuploid. And so in humans, aneuploidy is not a good thing. So why is it that in a strawberry it's very well tolerated and in a human it's very not? So we went back to a simple model organism, in this case the yeast, because we have a lot of power in this system. And we built what we call a ploidy series. So we start with a haploid genome, has 1 copy of all 16 of its chromosomes. We produce from this haploid a diploid. So these 2 strains are absolutely identical at every single position, except that one has 2 copies of its chromosomes and one has 1 copy. And then we made a tetraploid from that. And so these guys are completely isogenic, like I said, absolutely identical, aside from copy number. Oh, and one little other detail. And that is, some of them carry a yellow marker and some of them carry a blue marker. And what the yellow and the blue marker does is literally when we grow these in the lab, it turns them yellow or blue. Which turns out to be very useful from a technological standpoint, because there are machines that we can propagate these little yellow and blue guys and then feed that into a machine and it will just count the number of things that are yellow and the number of things that are blue. And what we can do with that counting information is, if during the course of just keeping these in the lab, say the yellow strain acquires a mutation that makes it less sick or makes it better fit to this condition, it will suddenly start to outgrow the blue. And so we're looking for those points at which mutations arisen that's a caused adaptation. So we grew our initial 3 strains under raffinose as a sugar source, which turns out yeast don't like raffinose as a sugar source, and said figure out a way to deal with this. And what I hope you see very quickly is that the haploids adapt at about the 150th generation, the diploids at maybe the 100th generation, and the tetraploids around the 40th. So something about having more copies of your chromosomes allowed for more rapid adaptation. Well, that was cool. So what the heck's going on? So we can sort these guys based on total DNA content, look at which chromosomes they retain, and what we see is that the tetraploids, what they were actually doing was going panic and starting to throw chromosomes out the window as they're driving down the street, right? And so they're all aneuploid. Aneuploid in different ways. Which chromosomes they kept, which chromosomes they gained, and I don't, how they gain is an interesting, whether that's biased in some fashion. And there were even small segments like this HST67 that they would gain just that little bit in copies. And so now we wanted to ask, what do these do? So this was a collaboration with David Pellman at the Dana-Farber Institute, and they did most of the evolution study. And they sort of came to us at the point of having the adapted clones and said, this is cool, but what's causing the adaptation and how is aneuploidy involved? And so we did our standard trick, and we threw them at a sequencer, and we analyzed 8 yeast genomes. The yeast genome's only 12 megabases. That turns out not to be too bad. Believe it or not, you can actually have a grad student who's really bored sit at a browser and just flip through it for about 3 months and get most of what you need. Which, I'm not going to say we actually did that, but when you get your predictions, the amount of false positives you have is non-trivial. So everything you predict does have to be looked at. So we initially predicted about 50 mutations per strain. And when you then, like I said, have an undergrad or grad student carefully look through those, it turns out that these are the only ones we could validate. So we got our 50 mostly false positives down to this strain had 3, this strain had 2 variations, and you see the tetraploids had considerably more. So one of the reasons tetraploids could adapt faster is by having multiple copies of the chromosomes. If they got a mutation and the mutation was recessive and problematic, they still had good copies of stuff to hang on to. And that's part of the reason we're diploid. It's a buffering mechanism. But it also allowed for them to acquire mutations more quickly, which allowed them to adapt faster. Now, we are not, when we get these lists of bona fide verifiable mutations, able to look at the list and say, how did they adapt? This is like Larry saying, there is no gene for dancing. But we can, in yeast, take one of these mutations we identified and then put it back in to the parent strain that's never been exposed to the growth condition, that didn't go through the evolutionary study, and say, does this actually help in this condition? And in this case, this is the SNF3 mutation. When we reintroduced it into the wild type, much better growth. In a diploid, much better growth. In a tetraploid, exceedingly great growth, implying that this thing is a dominant mutation, that a single copy of this variant suddenly makes you capable of living in this environment. We found a lot that weren't so useful because mutations are random. So then we transcription profiled all of these strains, sorted the transcription profiles on similarity, and we saw this very interesting graph. And what we find from this, after a lot of biochemistry and bioinformatics, is that all of these guys in the blue box acquired adaptation to this raffinose stress by a copy number variation of 2 genes, hexos 6 and 7, which are involved in a particular type of sugar metabolism. So that makes sense. These strains up here, let's just say they were a lot harder to figure out. The SNF3 mutation I just showed you is in this F12 strain. We have since identified in one of the haploids MTH1 that works in the same pathway. And so we would identify these mutations, and then to answer somebody's question that was addressed to Larry, we would go to that NAR database issue and start looking through that 1,500 databases for something that might be relevant to this particular gene to try to figure out why this might be helping with this particular kind of sugar. And I hate to admit this because we've now been working on this project for a couple of years. This strain right here, this one we call G11, started out tetraploid, lost many of its chromosomes. We can identify 4 or 5 mutations in it. We can reintroduce those mutations back into the parent, and so far we have no evidence of why it's capable of growing better in this sugar than its parent did. It's clearly acquired something, but even sequencing its genome and throwing everything we know about molecular biology at it, we still haven't figured it out. We will figure it out. Yeah. So, I'm not the only one using yeast to study aneuploid. Angelika Amon at MIT has done a ton of work on this. And some of the patterns that come out of Amon's lab and Rong Li's lab and make a few implications about aneuploidy. One is it has adaptive value. Another is that if you're aneuploid, you are a little stressed, possibly a lot stressed. And that stress induces in the cell a higher mutation rate. And that mutation rate then allows for you to, by chance alone, maybe happen into something that helps. And so the mutation rate in aneuploids are higher, but they rapidly adapt to being an aneuploid, which made us question whether or not that's happening in Down syndrome individuals. When that sperm and that egg meet and you end up with an individual that's trisomy 21, is their mutation rate for those first few cell divisions much higher? Maybe. That might explain why 75% of trisomy 21 individuals don't result in live birth. 75% are spontaneously aborted before live birth. It may also explain why, when you look at individuals with Down, the phenotypic variance is tremendous. At one end, you have the kids that are severely mentally affected, they have heart defects, they come down with celiac disease, they come down with Alzheimer's by the age of 12. Many of them don't live that long. And at the other end, you have the ones that have the classical Down's facial morphology and live to be 62. And we don't really understand that variance, but our hypothesis is it has to do with this early mutational change. And so we wrote a grant to the Linda Cernick Institute, which is one of the world's renowned research-based institutes on Down syndrome. It's actually based out of the medical school at Denver. And they're giving us money to do a pilot study on this to sequence a small number of Down's individuals. And just like with Lily Grossman, it was very informative to know which mutations were new in her genome relative to her parents to understand her disease. We need to know which mutations are new in this Down's individual relative to the parents. This is, let me back up and say, I get a little bit of funny looks from people when I say, oh, we're gonna sequence Down's individuals. They kind of look at me and go, pat me on the head and say, yeah, I know you have a degree in computer science. We know what causes Down syndrome. Down syndrome is just trisomy 21. That's not the point here. We're trying to figure out what is causing the variability in the Down's phenotype, and more interestingly, in those Down's individuals that are the healthiest, live the longest, and have the least amount of medical problems. Have they acquired a mutation that actually gives them that robustness? That's what we'd like to ask. That's what we'd like to assert. So this is a cool project, and I'll tell you the status of it. The status of it is that the International World Down Syndrome Congress is going to be held in Denver next month, and we're going to be there recruiting people to sequence. And despite saying that, if all works out in terms of our timetables and talking to reps from the sequencing companies, we should have 5 trios fully sequenced by October 1. So that also gives you an indication of the pace of genomic sequencing. So what I hope you've gotten today is that my lab's interested not in the bright lights so much as in the country, and trying to ascertain by comparing individuals within a species and moving more and more towards medically, how those things contribute to how our genome is read. So nothing's done in a vacuum. The mouse work was a collaboration with Richard Radcliffe. The Down syndrome stuff, or not Down syndrome, sorry, the yeast evolution with David Pellman. There are lots of other projects going on in the lab. It was mentioned in the nice introduction that my lab sort of studies 2 things. One's this individual variation, and the other's just transcriptional evolution and how these things arise and change. And there's all kinds of projects there in cancer and biofuels and biofermentation, because a really cool thing about asking fundamental biological questions is, like Larry said, you're like in all these disciplines at once. And so it looks really cool on your CV, even though you're really only doing 2 things. And so at this point, we'll take questions.
Larry, can you talk a little bit about how you see the role of the library and librarians in the work that you do?
It's really sensitive.
Then no. So I'm one of the, there aren't that many NIH investigators who put librarians on their research grants, but I'm one of them. I work with Lynn Fox at the Health Sciences Library. Boy, how to make friends and influence people. And so part of the work I do is text mining. And that means I have lots of issues with publishers and rights and stuff. You may think that when you buy access to these catalogs that you can do anything you like with them, but it turns out that that's not true. And so if I start citing all the things that we apparently have rights to and start running computer programs over them, they shut us off. I've had access to the, I've made my entire medical campus shut off from all kinds of different things. So we need help in rights management and stuff like that. So that's one way. We also, I think, really, well, I want to generalize a little bit from that. So in addition to the kind of things that librarians can provide, text miners, which is the specific sort of stuff, I think that an awful lot of researchers are in the situation that Robin and I are, where you get a result and something's known about this from some field that you've never picked up the first thing from, and you start reading those articles and it's like, what does this mean? Help me out here. And so another place to go is to try and, I don't know if the right word is get trained, but to find the resources it takes to understand what I need to understand. So that's another place librarians can really help.
I actually call it succumbing fear. Because when you're presented with a new field, your first impulse is always complete and utter, I'm an idiot here.
Run screaming for the door.
Yeah.
And then maybe a third way is in trying to understand reliability of resources. A lot of the stuff that I come to use in trying to understand these results that we get are not right out of the main databases that I showed you, or things that show up in nucleic acids research, and particularly in medical places where we're trying to understand complex phenotypes that patient groups may have been talking about, or trying to analyze social media, patient social media, things like that. Librarians are often helpful in picking out the right resources for us to use as well. So there's a whole bunch of different areas to add to that.
I actually want to give one anecdotal story on this. When I was a grad student at WashU, one of my colleagues was studying these weird hyperthermophilic bacteria and had made a bunch of predictions about their genome and wanted to go back and test them. And he checked out of the library book on how to culture these things. And as a grad student, you're allowed to keep a book forever, a month, 6 weeks. And he gets to the end and he doesn't want to turn it back because he's still working on his experiments. And so he contacts the librarian and he says, can I just recheck this back out? She says, no, it's been recalled. And he goes, recalled? Who else is doing this? And so the librarian put the two in touch with each other. It's a very obscure, very narrow area of research, only to find out that there was another grad student in the geosciences department at that university that was studying a very similar question. And the two of those guys hooked up, started having lunch together, figured out how to culture these things, and both of their research took off in ways that we never would have gotten. The interesting thing is that before my colleague talked to the librarian, because he thought there's no way the librarian's going to just tell me who this other person is, he put a note in the book and said, I don't know who you are, but please contact me. So, there's all kinds of ways that it works.
Do you guys ever go out into the public to show them the value of what you're doing so you can get more money for the grants with funding getting sucked up? Do you have tutorials or?
So I'm a horrible introvert. That said,
Never know it.
that said, I've spent a lot of time with just that. And next month at this World Down Syndrome Congress, we're literally spending 3 days in a convention hall in downtown talking to the parents of children with Down syndrome. And that's both very exciting and it's just incredibly scary. I will not sleep for a week before that conference because I can't explain what I do to my grandmother. So now I gotta explain it to people whose children are affected by the disorder, right? So I think part of the problem is that most scientists are geeks.
Yeah, but you made it funny, and that is a big thing.
And I
that
think a lot
helps
of
both
what's
of you.
important education-wise, that I know Larry shares this with me, with our students, is teaching them to talk, and teaching them to talk more than just their best buddy. And that's actually one of the hardest things for people to learn.
Yeah, so I also like doing lots of outreach. I mean, I think this is an example. We're here because we like doing that, right? And I do want to caution that while I think it's really important, and I also am a little bit hooked up with the rare disease community and patient communities and things like that, which is really satisfying in a lot of ways. It's not a way to get money. It really does not help with grant funding or other kinds of funding, for that matter. It's just, you got to do it because it's the right thing to do and because it's personally satisfying. I like. Right. You know, I'm hooked up with the art museum crowd here. There's the Denver Museum of Contemporary Art has this really cool series they call Mixed Taste, where they take two random experts from fields that have nothing to do with each other and then make them do something like Robin and I just did. Wow, so I talked about genomes, and I got hooked up with a guy who talked about muddy waters. And then we answered questions together afterwards, and we're not allowed to try and make connections until the question thing, and it's really fun. So we talked about blue jeans. But I think outreach is important for scientists. We are part of a training program that teaches computational biology, gives graduate degrees and the like, and we do talk about scientific communication. We mostly focus on communicating with other scientists across disciplines, which we have to do more than most scientists, but also that helps with people who communicate in the outside world. It's got to be a relationship at many levels. So some of us like, I love talking to the public. I think it's really fun. I was just on NPR last weekend. Oh, made me so happy. Right, and I could sleep like a baby before that. It was no problem. And some people just don't like it, and they, everybody should follow their talents and do what they think is right.
I have a question about, I noticed that you referenced in your talk that it was McGraw-Hill textbook, which I think might be OMBID, and that is something we license at my library, and I am a medical librarian. And then I also have, the biochemistry department that's changing all to more genetic kinds of research, and nutrigenomics and public health, so it's across every discipline. And it looks like the people at your level are using such specialized resources. And I'm just wondering how the library's licensing, how do you identify what's really going to be useful to, the people that are just starting out? Maybe they do need that. I was wondering, do we need OMBID, or do we need to look more at these more specialized things that will probably be not great for students in the master's program?
So when we get a new mutation and we have to chase it, and it's in a field that we've never been exposed to before, I often send the student first to go grab the most generic possible textbook in that field and become loosely acquainted with the terminology, the big concepts, because you're never going to be able to understand the more difficult literature without that first. But then they usually are very rapidly after that into sort of the detailed scientific literature. I don't really know if that addresses your question, but that's sort of the trajectory that most of my students end up embarking on.
But there's still an importance to have that traditional OMBID, which used to be a print book on the reference shelf, and now it's all on library plates. It's still important, yes, to maintain that as part of the library resources that we're offering.
Absolutely, because most of those in print are several $100s, if they even still print them.
Well, and I think that's right. One of the things that's happened with the sort of disciplinary breakdown that's happening in biology, and you know, it's not just us. The medical school is just beginning, our medical school in Colorado, a revamp of the basic science departments. And those basic science departments came into being mostly 40, 50 years ago based on the name of a class that we taught to doctors then. We don't do that anymore. We do an integrated organ-based curriculum, so the departments don't make any sense. And so we're going to tear them all apart, and maybe we'll just have one, or maybe we'll have four, but it's not going to be biochemistry and genetics, and you know, that's gone. And so part of the consequences of that means we all have beginner minds, right? There's no way you could know all of it. So whether from college students or even high school students through, you know, full tenured professors, if you're really going to be active in doing biomedical research these days, you find yourself in a situation where you've got to start from a pretty low level and learn something new. And it's one of the things that makes it fun, but it also means we need some of those more basic resources in the collection so that we can go get started and learn the basics of some new field quickly.
Larry, can you say a little bit more about what you do with text mining, what you're trying to do and how you do it?
Sure. I do lots of stuff with text mining. It's a fairly generic technology, but I'll tell you broadly, and then I'll give you a more specific example. So all those databases that I mentioned, those 1,500 databases, one of the things my lab does is try to integrate them using semantic web technology so that when you get a gene list of a couple hundred genes, we can pull on all those databases and try and help you put together that story and sort of synthesize some of that information together so you can do a more knowledge-based approach to analyzing your data. And those databases that we do that from are really incomplete. Okay, it just takes a lot of money and time to get the stuff out of the literature and into those databases. And so even the really good, well-funded ones are a year or two behind. And that's for the people who we want to use our system, that's devastating. You can't be a year or two behind. So we need to supplement what we get out of the databases with less accurate but broader and more timely information about what was published on that gene last week. And so that's the kind of main use for the text mining. But we have lots of specific projects where we can use the same technology for some other purpose. And my favorite ones are the ones that look through patient records, that look through medical records. So for example, we have a project that looks at lupus patients. So lupus patients, lupus is a chronic disease. It has flare-ups, and if you pay careful attention to the sort of how it unfolds over time, you can kind of get ahead of the flare-ups and treat it better. And so in order to be able to pay careful attention, physicians have developed a score that lupus patients get seen fairly frequently. You're supposed to calculate that score every time. And if you do that, you can see trends in those scores that can help you improve treatment. Physicians don't like doing it. It takes an extra 5 minutes. They think they already know how their patient's doing. They don't like actually putting the number in. They don't think it matters. So they still dictate the typical physician note, and we can use our text mining stuff to go through the note and calculate the score for them. And so that's a place where we can really improve patient care with a little text mining. And we have a similar thing for acute lung injury in neonates and stuff like that, where we try and use these techniques in some specialized application where we can do something with a free text, usually in the patient record, for our purposes out in the biomedical literature, but that has fewer applications unless we can make it work on a big scale. And we spend lots of time on supercomputing, and it's huge scale and fighting with publishers and stuff like that. One day I think that will really make a difference, but right now the real ones are mostly in patient application.
So in the HIPAA guidelines, limit what you can mine?
Amen, brother. So the NPR story was actually about privacy and the NSA and stuff like that. And the one place that we have the strongest privacy laws, it causes the most trouble. Okay, researchers cannot, the HIPAA, for those of you who don't know, HIPAA is the Health Insurance Portability Act, which also has in it privacy legislation, which regulates who can look at patient records. There are 3 people who can look at patient records, okay? The doctor taking care of the patient, that's a good thing, okay? So that helps with handoffs and stuff like that. The hospital or another institution that is part of the care of that patient, and the payer, that is your insurance company. Okay, those people can get access to everything. Okay, nobody else. The rest of us, Okay, so that leaves out the researchers who are studying the disease that you have. Okay, can't get at it. Need to individually consent each patient to look at research, to look at your patient record to see if we can get anything out of it. And that's a terrible thing, because if we could hook up patient records with genomes, we could discover a lot of stuff at very low risk to any individual patient, right? It's not like giving you a drug or a new surgical procedure or something. Kind of a shame.
And actually, our IRBs and other institute review boards that make sure that we're not doing anything unethical or potentially damaging to the individuals that we recruit explicitly require us to de-identify much of the information from our samples, which makes sense as a protective from the patient, but makes Larry's job 10 times harder and makes our ability to sort of do secondary analysis, essentially just takes it away.
Well, and I just had an editorial in Science, I don't know, whenever it was, 6 months ago, that talked about the damage it does to patients, because if we discover something incidental while we're doing research on your genome, we can't get back to you and tell you, okay, because it's been anonymized. So the way we get out from underneath this HIPAA stuff is we anonymize things. So we can't tell whose information it was. So that's how we get access to patient records that, so to write our lupus score, we get anonymized patient record data. Anonymization has some problems. So for example, one of the ways that HIPAA says you have to anonymize things is to take out the dates. And that by and large isn't that big a deal, except for if now we can't tell whether you got sick and we gave you a drug, or we gave you a drug and then you got sick. So it's kind of nice to have those dates, and there's tricks around trying to handle that. But it also means it's particularly hard to anonymize free text. So I can anonymize your lab values. That's pretty easy. All I have to do is take your name and your zip code and your birthday off of them. By the way, most people live in a zip code that has only 1 or 2 people who have the same birthday in it. So there's all kinds of ways to identify people you wouldn't have expected. But free text, you got to have somebody actually look at it to make sure the doctor didn't actually say Mrs. McGillicuddy in the text. Yeah.
I have a question about sort of the educational process for our graduate students, and this was kind of asked yesterday, but you're both computer scientists and then have these other disciplinary affiliations. Do all of our graduate students who are science majors need to be computer scientists as well now? And how does the education system address that or not address it? So I work with a lot of graduate students here. I'm an environmental scientist, but now I'm learning Python, and I have no support for having to do that. What do we do about that?
Could you repeat the question?
Yeah. So the question was, both Larry and I have computer science backgrounds, and we do this broad interdisciplinary work, and a lot of these disciplines have a lot of data associated with them. So on some level, does everybody need to have a computer science degree in their background to make progress in these? So it's a question about the education initiatives system. So I can talk about one thing that we've done here. So as part of the Biofrontiers Institute, we established a new graduate school program called Interdisciplinary Quantitative Biology, which has the cute acronym of IQ Bio. In the IQ Bio program, we bring students in, and they are completely unaffiliated with any department on campus for a year. And they take a series of courses that are taught project-oriented. So here's a cool problem in science, medicine, biology, and here's all the different ways that you can approach it. And so there's a lot of engineering, there's a lot of math, there's a lot of chemistry, there's a lot of biochemistry, there's computing. And our expectation is that this cohort, and it's a small cohort, it's 8 to 12 students, and they come from different backgrounds, will by sink or swim, basically, and work with each other to figure out how to learn and solve these problems in these problem-based classes. And then they take rotations with various individuals on campus, and at the end of the first year, they all sort of raise their hands and say, okay, I want to be a computer science major, I want to be a bio major, and then they sort of enter into a traditional graduate program. But it's a way for them to get exposed cross-discipline that is not typical in a normal educational environment. So that's kind of cool, but it doesn't really address your question. I personally think that molecular biology, by definition, is going to be a computational science within a decade. And that doesn't mean that everybody that studies molecular biology will have a computer science degree, but the biology major will have to have more quantitative and more computational concepts integrated into it at all levels, all the way down to through the undergrad. And I'm loosely involved with a number of initiatives trying to get that working here.
So I'm going to take a slightly different tact on that and try and tease apart what computer science means a little bit. Because I don't think...
It's like Gene.
Yeah. Yeah, that was great, by the way. I totally agree with you about Gene. Computer science as a discipline is really about how we can draw conclusions about computational phenomena, how we can study computational phenomena. And so mostly scientists don't need to know that. Okay? They don't need to understand the way that computer scientists prove theorems about computational complexity, say. So that's sort of computer science as an academic discipline. Then there's programming, which is the ability to sit down and write computer code that will do something. And so when you say you're learning Python, you're learning how to program. And that's different than computer science. There are people who are good programmers who don't know any computer science. There are computer scientists who can't program, okay? They're theorists mostly. They work with pencils still. They prove theorems. And so programming may be useful to people who are doing other kinds of science. Depends on what exists to do your thing and if the lack of some tool is getting in your way. But programming is a thing that you have to practice. I like to analogize it to learning to play an instrument, okay? You can't just read a book and remember it and know how to play an instrument anymore. You can read a book and remember it and know how to program. You got to do it. It's a practice. And it takes time to become good at it. And not everybody can, kind of like a musical instrument, right? Okay? I'm just never going to be a great musician. I can kind of plink away at some things, and that's useful occasionally. And that's a good metaphor for programming, okay? So sometimes it's useful to be able to plink away a little bit at a computer and write a little script that will do something because nobody else has done it. There we go. But by and large, not everybody has to be a great musician and not everybody has to be a great programmer. And the reason that's true, you can make progress in science, is because you get sophisticated about how to use computational tools that aren't programming. Okay, and this is something that's just coming everywhere, okay? My kids are really pretty good at using computers to get what they want, and they can't program a line. Well, my son a little bit, but they're sophisticated users. They know what's out there, they know how to take advantage of it, they know how to put pieces together, and they know where to go for help. And so I think that last bit, making biologists or other kinds of scientists, environmental scientists, sophisticated computer users, I think that has to happen. That's got to be part of the educational system. Programmers or computer scientists, not so much, not for everybody.
I agree.
Yes, sir.
Could you talk a little bit about the data aspects of how you crank these data and get results out of it? Like, how do you process that? Like, is it a lot of noises, and how do you preserve these data, sharing this?
Yeah.
Yeah, but so you're the one who deals with the sequencer output.
Yeah, I do a lot of sequencer output. So we literally, one of these projects, for example, the mouse sequencing one, like I said, is terabytes of data, raw data. We find that, so we get the raw data, it comes in those little 50 base pair reads, you have to check those for quality, you have to map them back to the reference genome. There are multiple steps just in what I would call quality control before you ever get to where you're doing anything interesting. Many of those steps, we use open source tools. So we're very strongly dependent on the open source software community, and we contribute back to the open source software community anything that we develop. Then we get to sort of the analysis stage, where you're asking questions nobody's asked before, and often that requires building some new tool. And what we have found is between the point the data comes off the sequencer and the point that we're starting to write up a paper, we get about a 20-fold increase in the amount of stuff on the hard drive, intermediate files, attempts to try something that doesn't work, graphs, plots, etc. So if you think about a project that has 10 terabytes of data, and we've just increased that by a factor of 20, that's why we have big machines that can heat a room. It's not that they're actually doing a lot of compute, it's that they're storing a ton of data. So to publish, traditionally, you've had to submit anything you sequence to one of the national repositories. But sequencing has now gotten so cheap that the repositories are literally crumbling under the weight of people submitting terabytes of data a day to them. And they just don't know really how to store it, how to deal with it. So that's actually a huge, I think, problem moving forward. There are some editorials out there about people that are saying very quickly the cost of sequencing is going to get to the point that we no longer store it, we sequence it, throw it away, and then if you need to know what it was, you'll just resequence it. We're not there. We're not going to be there for a little while because the analysis end is not going to be able to keep up with that kind of real-time processing. But these data sets are not small. So Larry and I both have backgrounds in machine learning, and one of the basic tenets in machine learning is you take a giant data set and you look for patterns in it. And you know, if you want to boil, this is the best thing I've got for a grandmother description too. If you want to boil down what we do to one sentence, is we look for patterns. And to do that, you got to have a ton of data.
Yeah, I'm sort of lucky in that I live the much more processed end. So my world is about taking a big gene list and trying to help people make sense of it. And so a big gene list has 200 gene names in it. That's not an enormous amount of data. Now it took going from the terabytes through all that computation to get to that couple hundred gene names, or polymorphisms or proteins or whatever they turn out to be. And our big data actually comes from the literature. So we take the entire full-text biomedical literature, as much of it we can get publishers to give us, and run text mining programs over that. And we take all 1,500 of those databases, some of which are really enormous, and we don't take all of them yet, and try and suck them into one unified representation that's semantically well-founded. So we tend to deal with symbolic data rather than sequence data. That has its own challenges, and we also have a similar kind of supercomputing need to the sequencers that's different than the physicists. So the physicists, I like to joke, the physicists take one big matrix, they load it into a computer, they push compute, it computes like crazy for 3 weeks, and at the end they get a number, okay? The answer, 42, whatever it is, right? We don't do that, okay? We take a terabyte of data that takes so long to move it over the internet that it's often the case that I put the disk in my car and drive up here rather than trying to move the data over the wires because it's so much faster.
So I have to interrupt you there because you said you brought down the medical school computer system. I've brought down the medical computer systems just trying to move data from their sequencers up here.
Yeah, I got into trouble for a similar kind of thing.
Yeah.
And we actually did this. I have a collaboration going with the Pacific Northwest National Laboratory and to semantic supercomputing, and so we wanted to give them our knowledge base, and we calculated it would have taken like 3 months of the entire internet connection between here and PNNL to move it. So we put it on a big disk and brought it. No, he came to Rocky. Came to this conference I run in Snowmass every December, and we handed the disc to him, and it felt like something out of a spy movie. You know? It was really, here's the data. It was hilarious. But anyway, so we have different issues than the physicists in supercomputing. Most of our stuff is about very large memory computations. So in order to be able to align those bits of sequence to that reference, you need to get them all into memory, because if you're constantly going back out to disk, to find it slows everything way down. You can't do it. So both our kind of text mining and semantic computing stuff need very large memory, and the sequencers need large memory. So we just got a shared instrumentation grant from NIH to build a special kind of supercomputer that's really all about input-output bandwidth, so moving stuff in it fast and getting stuff out of it fast, and memory. And it's not like, and the compute is not all that special. It's not, the physicists wouldn't be interested in what we have. But that's the kind of computing we have to do, and it's really all around data. And also the kind of compute we do when we suck in all that data and compute on it a bunch, what we're outputting is more than we put in. Okay, it's bigger than what we put in. So it really is about moving data around through computers, and it's sort of a technical thing. It's not a big data in the sense of, we need a refrigerator-sized thing to put it in. But it is very different than traditional supercomputing loads, which have really been about complex calculations rather than about calculating fairly simple things over a really large amount of data.
Well, thank you.
Yeah, I think they're hungry.